So DNA contains the information that a cell needs to carry out all of its functions. So in a way, DNA is kind of like a cell's encyclopedia. Suppose that you go to the library to do research for a science project. You find the information in an encyclopedia. You go to the desk to sign out the book, but the librarian informs you that the book is for reference only and may not be taken out. If it's a reference only book, why do you think the library keeps those books for reference only? You can't check them out. Probably because they're too important, right? They're usually large volumes of information. So if you can't borrow a book, how can you take home the information in it? Well, you probably have to make a copy of it somehow. And so by making a copy of it somehow, you'd have the opportunity to take that information with you, right? You can't take the original, but you can take the extra copy. So all of the parts of a cell are controlled by the information in DNA, yet DNA cannot leave the nucleus. It's maintained inside that nuclear membrane in order to protect it and prevent it from mutating. So how do you think the information in DNA might get from the nucleus to the rest of the cell? It's an interesting question. It's kind of like the reference book. We're going to have to make a copy of it. So, this process is called transcription. When you take an original copy and you handwrite notes or you transcribe the information from it, that is called a written transcription, right? There's other types of transcriptions that could be done, okay, but that's just one example. Now, RNA is the molecule that is going to transcribe that information found in the DNA. RNA carries the genetic information from the nucleus to the ribosomes. It uses coded information then to build proteins. RNA differs from DNA in that RNA is single-stranded. Remember, DNA is double-stranded. RNA is only one side of the ladder. It almost makes it look like a comb. RNA has ribose sugar instead of deoxyribose. So RNA's name is ribonucleic acid as opposed to deoxyribonucleic acid. Now, the big major difference is in the language, so to speak, of the nucleic acid. RNA has a base called uracil, so it has a U instead of thymine. There is no thymine found in RNA. So that means uracil pairs with adenine. That's the complementary base pair in RNA. So that's one of the major differences, and that's why RNA is able to actually carry a copy of the information in the DNA out of the nucleus, okay? So mRNA is one kind of RNA that's used in this process, and it's called messenger RNA. It literally is the messenger. It's like a bike messenger, taking the copy from one place in the cell to another. That's pretty much its role. It transcribes, makes a copy, and takes it out to the cytoplasm, out to the ribosomes. The tRNA is known as transfer RNA, and it brings amino acids to the ribosomes and puts them in order. Okay? I like to think of the transfer RNA as kind of like a construction builder. Okay? They're the ones that they don't have the master plan, but they go through and they work individually, whether they're plumbers, electricians, or carpenters, it doesn't matter. Everybody does their job in order to make that building. It's the same thing with the transfer RNA. Different pieces of transfer RNA have different jobs, and I'll explain that more in depth in a little bit, but it's more that they each do their part to make that protein. Then there's ribosomal RNA. And at this point, scientists don't know that ribosomal RNA has a real specific job. All they know is that it's located literally in the ribosome. So it helps make up that structure of the ribosome. Now, transcription, this whole process, is how the DNA gives its code or transfers a copy of that gene to the RNA. Okay? So it's kind of like DNA replication in where the DNA unwinds and unzips, and we're going to take a look at some diagrams here in a little bit. Using DNA as a template, free RNA nucleotides come in and match up with the complementary bases. So it's the opposite of, I'm sorry, not the opposite, but it creates those complementary base pairs. Sorry, the dog came running in here and I lost my concentration. So RNA polymerase is the enzyme that literally links those RNA strands together, okay? The RNA strand gets released, the DNA re-zips up like a zipper on a sweatshirt, okay, like a hoodie. You unzip it, that's where replication as well as transcription can occur. At the end, it re-zips up again and then rewinds. So the DNA is left completely intact, unharmed, unchanged, and the RNA, the messenger RNA now, is a copy of whatever gene was being transcribed. It leaves the nucleus through the nuclear pores and goes out to the cytoplasm, out to the ribosomes. So remember, the order of the bases in DNA is what carries the code. The RNA complements the DNA, so it also carries the code. So let's take a look at exactly how this works. So inside that nuclear membrane, remember there are pores, and those pores enable the RNA to pass through, but not the DNA. Double-stranded versus single-stranded here. So, whoops. Oh, hello. How in the heck did I just do that? So, sorry about that. So we've got the RNA polymerase enzyme here going in and reading the DNA at the replication fork, okay? Remember, at this point, it's not called a replication fork necessarily. I'm just using that term to help you refresh your memory. But where the DNA unwound and unzipped is at what's called a promoter region. And that promoter region indicates where the RNA polymerase comes in and starts reading it. It starts forming that single strand of RNA, okay? So let's take a look at exactly how DNA transcription works. Now, I think I've got the videos working this time, so let's see if it works.
Instructions for making proteins are coded by segments of DNA called genes. But proteins are not made in the nucleus. Instead, a strand of ribonucleic acid, or RNA, must carry the instructions for making proteins out of the nucleus. This type of RNA is called messenger RNA. Both DNA and RNA are long chains of nucleotides. Each nucleotide contains a sugar, a phosphate group, and one of 4 bases. RNA is different from DNA in 3 important ways. First, RNA is a single strand. Second, RNA has a different type of sugar called ribose in its nucleotides. Third, RNA contains the base uracil instead of thymine. The other bases found in RNA are the same as in DNA. They are cytosine, guanine, and adenine. The manufacture of RNA from a DNA template is called transcription. Transcription begins when the 2 strands of DNA in a specific region unwind and separate. Only one strand of the DNA serves as a template for the new RNA. The enzyme RNA polymerase matches RNA nucleotides with the complementary ones on the template strand of DNA. Recall that RNA contains uracil instead of thymine. Therefore, uracil pairs with adenine on the DNA strand.
That's important. Check that part out.
C is paired with G. A is paired with T on the DNA strand. And U is paired with A in RNA.
That's the big difference.
As the nucleotides are assembled in the proper sequence, RNA polymerase moves along the DNA strand, assembling the messenger RNA, until the entire gene has been transcribed. Hydrogen bonds reform between the complementary bases in the DNA molecule, reattaching the 2 strands. The coded instructions on the DNA molecule have been copied to the newly manufactured strand of messenger RNA.